Good afternoon, everyone. Myself, Dr. Somdat Mujwar. I am working as an associate professor in MMCP, Maharshi Manakandeswar University, Mulana. The topic for today's lecture is beta-lactam antibiotics. Beta-lactam antibiotics are basically a class of antibiotic drugs which are characterized by a structural feature that is known as beta-lactam ring in their chemical structure. The beta-lactam ring is a 4-membered heterocyclic ring that is having a nitrogen atoms and a ketone group. The beta-lactam antibiotics inhibit the synthesis of bacterial cell wall. The beta-lactam antibiotics are supposed to have different antibiotic drugs. The common examples including penicillins and cephalosporin. The first structure is of penicillin, which is characterized by a fused 5-membered heterocyclic ring in its structure, and the second example is cephalosporin, which is having a fused 6-membered ring to this particular beta cyclic structure. So basically these two beta lactam antibiotics are supposed to have a bicyclic structural characteristics that is known as penicillinic acid in the case of penicillin antibiotics, and in the case of cephalosporin it is called as cephalosporinic acid. In case of penicillin, the beta-lactam ring is fused to a thiazolidine ring. This particular 5-membered heterocyclic ring characterized by the presence of a nitrogen and sulfur atom is called as thiazolidine ring. That is the structural feature of penicillin antibiotic. Secondly, in the case of cephalosporin, it is the beta-lactam ring is fused to a 6-membered heterocyclic ring that is called as dihydrothiazine ring. The dihydrothiazine ring is characterized by the presence of a double bond. Mechanism of action: all the beta-lactam antibiotics are supposed to interfere with the synthesis of bacterial cell wall. The bacterial pathogen synthesizes the UDP N-acetylmuramic acid and UDP N-acetylglucosamine, which links together to form long strands by splitting of the UDP molecule. The final long chain structure is stabilized by the formation of NAG-NAM alternative chains. The final step is the separation of terminal D-alanine by the enzyme of transpeptidase for the formation of cross linkage between these neighboring chains. In this way, in the given figure, it is clearly visible that the chain having NAG and NAM molecules alternatively is a part of bacterial cell wall. The NAG-NAM chain combines parallelly with each other by the synthesis of a peptide linkage. This particular peptide linkage is synthesized by with the help of an enzyme that is known as transpeptidase. So we can basically say that this particular transpeptidase enzyme is the responsible for the synthesis of transpeptidase linkage, that is responsible for adjoining the different chains of NAG and NAM responsible for the synthesis of cell wall. The penicillin antibiotic, or broadly we can say that the beta-lactam antibiotic inhibits the transpeptidase enzyme so that the cross linkage does not takes place, and therefore no stability or rigidity was maintained in the bacterial cell wall. This particular peptide linkage that is responsible for the joining of different chains together is responsible for generating the desired strength of the bacterial cell wall. And in the case of beta-lactam antibiotics, the inhibition of transpeptidase enzyme inhibits the formation of this particular peptide linkage, resulting in the formation of deformated cell wall, or we can say that defective cell wall, which is not having the desired strength which is required for the protection of the bacterial components from the external forces or external environment. So the transpeptidase and the related proteins are also known as penicillin binding proteins and are present in every bacterial cell wall. So we can say that this particular penicillin binding proteins are the part of almost every cell wall, and so these particular target proteins are targeted by this particular penicillin antibiotics or beta-lactam antibiotics in the broad sense. So penicillins is a general term which is used for the entire group of natural and semi-synthetic penicillin. So penicillin is characterized by the presence of this particular bicyclic ring structure, that is having a beta-lactam ring that is fused to a 5-membered ring structure. The prototype drug of penicillin class is penicillin G, or we can also call it as benzyl penicillin, which is characterized by the presence of this particular benzyl group in the amide substitution at 6th position. Penicillin was firstly discovered antibiotic which is still one of the most effective systemic bactericidal agent against gram-negative as well as gram-positive bacteria. So it is a bactericidal antibiotic which is having bacterial killing effect on the pathogenic microorganism, pathogenic microorganism. So it was the one of the firstly discovered antibiotic which was supposed to be accidentally discovered by the scientist, well-known scientist Alexander Fleming, right? And it is one of the most utilized antibiotic till date. It was initially obtained from fungus that is known as Penicillium notatum, but the present source is Penicillium chrysogenum. So we can say that the discovery of penicillin is performed. Firstly the penicillin was discovered from the fungus Penicillium notatum, but nowadays it is obtained from Penicillium chrysogenum. All the natural penicillins are derivative of 6-amino penicillin. We can clearly see that the structure of penicillinic acid, the penicillinic acid is characterized by the presence of a beta-lactam ring in the left hand side of the figure, and having a thiazolidine ring in the fused to this particular beta-lactam ring from the right side. It is the numbering of this beta-lactam, this particular penicillinic acid starts from this particular sulfur of this particular bicyclic ring structure. The sulfur is given first numbering. The adjacent carbon having 2-methyl substitution, dimethyl substitution, is numbered second. Then this particular third carbon atom is having a carboxylic group. Nitrogen is numbered 4. Then now we again comes to the 5th numbered carbon, that is the present in the junction of these two rings, and then 6th carbon atom is having amino substitution, or basically we can say that it is having an amide substitution, and this ketonic group is represented by 7th position. So penicillin nucleus is characterized by the presence of thiazolidine ring which is fused with beta-lactam ring. So this bicyclic structure is the structural characteristic feature of this penicillin nucleus, and both these rings combinedly together called as 6-amino penicillinic acid, having amino substitution at 6th position. The side chains are attached by a amide linkage. This amino group is preferred to be present in the form of amide linkage and having a R substitution. This R substitution is represented by using this particular R group. The substituent R determines the antibacterial spectrum of this particular penicillin antibiotics. Also it regulates the gastric stability or stability of the antibiotic drug against the gastric hydrochloric acid. Now we next move to the classification of this particular penicillin antibiotics. Firstly, the penicillin antibiotics are classified on the basis of their origin. Either they are obtained from natural resources, or they are derived from they are semi-synthetically derived from natural penicillin. Firstly, we study about the natural penicillin. The example is benzyl penicillin or penicillin G. This particular benzyl penicillin is characterized by the presence of R substituent in the form of benzyl group, that is CH2C6H5. This particular R group is clearly visible in the form of benzyl substitution. Secondly is second class is semi-synthetic penicillin. The semi-synthetic penicillins are supposed to be derived from this natural existing penicillin, that is benzyl penicillin, and all the penicillin drugs are the analogs or derivatives of this particular natural penicillin, that is benzyl penicillin. The different derivatives are formed, different derivatives of this particular benzyl penicillin are prepared with intention to increase its potency and improve its pharmacokinetics. These semi-synthetic penicillins are further subclassified into first class, that is penicillinase resistant penicillin. The penicillinase enzyme is responsible for the metabolic degradation of penicillin antibiotic. These are synthesized by the pathogenic bacteria. So first example of this particular penicillinase resistant penicillin are oxacillin, and oxacillin is characterized by the presence of this particular ring substitution, that is a heterocyclic ring having oxygen and nitrogen, and this particular isoxazole is substituted with a methyl substitution and a phenyl substitution. This is the structure representation of oxacillin. The second example of penicillinase resistant penicillin is floxacillin. Floxacillin is again represented by the presence of R substituent in the form of this substituted isoxazole ring having a methyl substitution and a substituted phenyl, chloro and fluoro substituted phenyl ring, which are having chloro and fluoro substitution at both the ortho positions. Now we comes to the second subclass of semisynthetic penicillin, that is aminopenicillins. Aminopenicillins are characterized by the presence of benzyl substitution having benzyl functional group, amino substituted benzyl substitution, amino substituted benzyl functional group. This is the structure of ampicillin. The example of aminopenicillin, first example of aminopenicillin is ampicillin, which is characterized by the presence of this amino substituted benzyl functional group. And second example is amoxicillin. Amoxicillin is characterized by the presence of para-hydroxy benzyl substitution having a amino substitution at the first carbon atom. These are the examples of aminopenicillin. Next we move to the third subclass of this particular semisynthetic penicillin, that is anti-pseudomonal penicillin. Example is the first subclass. This anti-pseudomonal penicillin are further subclassified into carboxy penicillin, characterized by the presence of a carboxyl group. This carboxy penicillins, the example of carboxy penicillins are carbenicillin, which is characterized by the presence of a carboxyl substitution at the alpha carbon atom, and ticarcillin is characterized by the presence of carboxyl substitution having a 5-membered heterocyclic ring, that is thiophene. The example is ticarcillin and carbenicillin. Next is uridopenicillin. Uridopenicillin is characterized by the presence of urido group, that is NHCONH2, and the examples of uridopenicillin is azlocillin and meslocillin. The azlocillin is characterized by the presence of substituted amino group with this particular urido heterocyclic system, and meslocillin is again characterized by the presence of substituted amine group with this urido heterocyclic system having a sulfur dioxide group. Now we move to the next class, that is acid resistant alternative to penicillin G. There is the prototype drug of this particular class penicillin antibiotic is benzyl penicillin, and the main problem associated with the use of this particular benzyl penicillin is that it is easily degraded by the gastric hydrochloric acid. It is not that much stable against the gastric hydrochloric acid, so that it cannot be administered by oral route. So by the structure activity analysis, the penicillin V is developed, semisynthetic penicillin V is developed from this particular penicillin G, which is supposed to be effective against supposed to stable against the gastric hydrochloric acid. So it can be administered by oral route as well. This is characterized by the presence of this particular oxygen bridge between this particular phenyl group and this methyl substitution. Right, and the next class is miscellaneous, that is the only example belonging to this particular miscellaneous class is azidocillin, that is characterized by the presence of the azido group. This is N double bond N is the azido group, which is the characteristic of this particular azidocylene. This is the only structural functional moiety that is present, that is the main structural substitution in the case of with respect to benzyl penicillin, right. The presence of azido group is the structural characteristic of this particular azidocylene. And now we can also classify this particular penicillin antibiotic based on their spectrum of activity. The first one is narrow spectrum penicillin antibiotics, second one is extended spectrum penicillin antibiotics. So narrow spectrum antibiotics includes the examples like penicillin G and penicillin V. The penicillin V is supposed to be stable against the gastric hydrochloric acid, so we can administer this particular penicillin V by oral route, and penicillin G is the prototype drug of this particular class, that is benzyl penicillin, and it is supposed to be unstable against the gastric hydrochloric acid. Second class is extended spectrum. These extended spectrum penicillin antibiotics are further subclassified into aminopenicillin. Examples are ampicillin and amoxicillin. We have already discussed about the structural feature of the ampicillin and amoxicillin. Ampicillin is characterized by the presence of a substituted amino group in benzyl penicillin, and the amoxicillin is characterized by the presence of a hydroxyl group in the benzyl penicillin, a substituted amino group as well as a substituted hydroxyl group in the para position of the phenyl group. Next we comes to the second subclass, that is carboxy penicillin, that is characterized by the presence of a carboxyl group, carboxyl substitution. Examples are carbenicillin and ticarcillin. In ticarcillin there is a substituted thiophene ring in the presence of phenyl substitution. And third one is uridopenicillin, that is the examples of uridopenicillin is azlocillin and meslocillin. All these classes are supposed to having extended spectrum of activity as compared to its natural congener, that is penicillin G, and its gastric acid stabilized penicillin, that is penicillin B. Let us move to the next class, that is penicillinase resistant penicillin. This is the members of this particular class are supposed to be stable against the penicillinase enzyme, which is synthesized by certain pathogenic bacteria. The example belonging to this particular class is oxacillin and floxacillin. Now we move to the structure activity relationship. And the first point is that the beta-lactam ring is very essential for the activity. So we cannot perform any kind of modification or structural changes or structural alteration in this particular beta-lactam ring. So we have to keep this particular beta-lactam ring as intact. And the second point is the amide side chain is essential for the activity. So it determines the stability of penicillin against degradation by gastric acid and penicillinase enzyme. This amide side chain is very important for the activity of very important for the antibacterial activity or antibiotic effect, activity, because this particular functional moiety, this particular amide group regulates the stability of this particular penicillin antibiotics against the gastric hydrochloric acid as well as the penicillinase enzyme, which is responsible for its metabolic degradation. By increasing the steric hindrance at alpha carbon atom of the acyl group, this is the acyl group, NHCO is the acyl group that is present in, that is substituted in the sixth position, sixth carbon atom of the bicyclic structure. So this particular acyl group is responsible for its stability against acid hydrolysis. So it cannot be degraded by the gastric hydrochloric acid. So this particular position alpha carbon atom, that is the next carbon atom to the this acyl group, is responsible for the stability against the gastric hydrochloric acid, hydrolysis by the gastric hydrochloric acid, as well as this particular site is also responsible for the metabolic degradation by the beta lactamase enzyme. So we can increase the steric hindrance by using some bulkier groups, nearby bulkier groups, so that this particular carbon atom becomes sterically hindered to get metabolized by either by the acidic hydrolysis or either by the effect of beta lactamase enzyme. The example of the example of the penicillin antibiotic which is supposed to having steric hindrance in this particular alpha carbon atom of the acyl group is methicillin, which is characterized by the presence of 2 methoxy groups at both the ortho positions. Penicillins are not suitable for oral administration because they are supposed to have poor solubility characteristics, but when their salts are used, the problem can be resolved as the salts are having good solubility characteristics, so they can easily be absorbed by oral route. Otherwise, the penicillins are having poor bioavailability after oral administration because they are supposed to have poor solubility features, as well as they are easily degraded by the gastric hydrochloric acid. Sulfur of the thiazolidine ring can be replaced with oxygen or carbon with increased activity as well as spectrum. The sulfur atom, that is the at first position of the ring structure, bicyclic ring structure, can be replaced by either oxygen or carbon, and the resultant compound formed by the substituting this particular sulfur with oxygen and carbon atom is supposed to have increased activity as well as increased spectrum of activity. So, the resulting compound is supposed to have the broad spectrum of activity as compared to the normal penicillin antibiotics. The N4, the fourth atom, that is the nitrogen atom at ring junction, is vital as it contributes to the reactivity of beta-lactam carbonyl center. The the nitrogen atom present at the fourth position, that is ring junction, is very important for the antibiotic activity because this particular nitrogen atom is responsible for the maintenance of reactivity of the carbonyl center present in the adjacent carbon atom or adjacent position or nearby carbonyl center. So, this we cannot do any kind of substitution at this particular nitrogen atom that is present in the ring junction or at the fourth position. The acyl side chain can be replaced with hydroxymethyl group. This particular acyl side chain can be replaced with hydroxymethyl group with increased activity against gram negative species. This substitution is supposed to increase the spectrum of activity of the existing penicillin antibiotic against the gram negative bacteria. The ester of carboxyl group at C3 position, the C3 carbon atom is having a carboxyl substitution. This carboxyl group can be substituted, can be modified to it the ester products that is supposed to have increased lipophilicity, and this particular increased lipophilicity can increase the spectrum of activity against the certain gram positive bacteria. Example is acetomethyl ester derivative. We can replace this particular carboxyl group with acetomethyl ester derivatives in the form of prodrug. The introduction of C6 alpha methyl group, the introduction of alpha methyl group at C6 position results in greater stability against the beta lactamase without significant loss of potency. The introduction of this particular alpha methyl group increases the steric hindrance for the in the nearby carbon atom, resulting in the increased stability against the beta lactamase enzyme that is responsible for its metabolic degradation. The substitution of electron withdrawing group at alpha position of acyl group increases the resistance to acid hydrolysis and therefore its overall stability. So, we can also substitute the alpha position of the acyl group with some electron withdrawing electronegative atoms, so that this particular the resulting compound will become more stable against the acidic hydrolysis, right. So, that its stability can be further. The general uses of penicillin antibiotics, being in broad spectrum antibiotic, penicillin class are used for the treatment of multiple type of infections in human body. The common uses are for the treatment of urinary tract infection as well as for the treatment of respiratory tract infection. Thank you.